Dear children, today's topic is ferritin, storage of iron. Myself, Dr. DIGNA VARGHESE. Ferritin. Ferritin is a universal intracellular protein that stores iron and releases it in a controlled fashion. Ferritin is the principal store of non-heme iron in animals. Most iron is occupied in hemoglobin and myoglobin that we know. The protein is produced by almost all living organisms, including archaea, bacteria, algae, higher plants, and higher animals. It is the primary intracellular iron storage protein in both prokaryotes and eukaryotes, keeping iron in a soluble and non toxic form. In humans, it, it acts as a buffer against iron deficiency and iron overload. Ferritin is found in most tissues as a cytosolic protein, but small amounts are secreted into the serum where it functions as an iron carrier. So, ferritin is found as a cytosolic protein in almost all tissues. Cytosolic protein means the protein molecules that do not bind to the cell membrane. Ferritin in small amounts are secreted into serum. So, where it functions as an iron carrier. So, in serum, ferritin acts as an iron carrier, but its most important function is it acts as a storing unit for iron. Plasma ferritin is also an indirect marker of the total amount of iron stored in the body. Hence, serum ferritin is used as a diagnostic test for iron deficiency anemia. So we can test how much iron is in serum, and it is a diagnostic test for iron deficiency anemia. Okay. Aggregated ferritin transforms into toxic form of iron hemosiderin. What is hemosiderin? Hemosiderin is an iron storage complex that is composed of partially digested ferritin and lysosomes. The breakdown of heme gives rise to biliverdin and iron. Okay, the body then traps the released iron and stores as it is a hemosiderin in tissues. Hemosiderin is also generated from the abnormal metabolic pathway of ferritin. The iron within deposits of hemosiderin is very poorly available to supply iron when needed. So it's a toxic also. Here you can see the brown colored one is the hemosiderin. So aggregated ferritin transforms into the toxic form of iron hemosiderin. Why it is toxic? The iron within in the hemosiderin, deposits of hemosiderin is very poorly available to supply the iron when needed. Ferritin is a globular protein complex consisting of 24 protein subunits forming a hollow nanocage with multiple metal protein interactions. Ferritin that is not combined with iron is called apoferritin. Ferritin that is not combined with iron is called apoferritin. Ferritins have two compartments, a mineral core that contains up to 4,500 iron atoms and a protein shell. So, ferritins have two compartments. One is a mineral core and the other one is a protein shell. The mineral core contains 4,500 iron atoms. Apoferritin, that is the protein shell devoid of iron, can be prepared by the treatment of ferritin with reducing agents and an iron 2 chelating ligand. Okay, so apoferritin can be prepared from the combination of ferritin plus a reducing agent and iron 2 chelating ligand, that is apoferritin without iron atom. Apoferritins have average molar masses in the range of 460 to 550 kg per mole. The protein shell consists of 24 subunits that link together to form a hollow sphere with the two-fold, three-fold, and four-fold symmetry axis. We know in a ferritin there are two compartments. One is a mineral core and the other one is a protein shell. Mineral core contains 4,500 iron atoms. The protein shell devoid of iron is known as apoferritin. Apoferritin can be obtained by treating the reducing agents with ferritin and an iron 2 chelating ligand. Apoferritins have the molecular mass about 460 to 550 kg per mole. And the next component of ferritin is the protein shell. Protein shell contains or consists of 24 subunits of that link together to form a hollow sphere with two-fold, three-fold, and four-fold symmetry. The threefold and fourfold symmetry axes of apoferritin are hydrophilic and hydrophobic pores respectively. We know the threefold channel is hydrophilic one, so it is most suitable for the ions, metal ions. But the fourfold channel is suitable for the hydrophobic pores and which is suitable for the protein molecules or lipid molecules. Okay. So the threefold axis pores are suited for the passage of ions. The most feasible mechanism so far proposed for the reversible incorporation of iron in ferritin involves its transport in and out as iron 2, which is soluble in and at neutral pH, that is at 7, but more likely some type of chaperone complex. Chaperones in molecular biology, molecular chaperones are proteins, okay, that assist the conformational folding and unfolding and assembly or disassembly of other macromolecular structures. So we know ferritin is a storage unit of iron, but the most feasible mechanism was the reversible incorporation of iron in ferritin involves its transport in and out as iron 2. Okay, iron 2. The cell is or ferritin cell. The threefold channel can transport the iron as iron 2 in ferritin, which is either it is a soluble at neutral pH, but more likely some type of chaperone complex. Okay, that can be complex with some proteins. If it is a protein, it can definitely pass through the fourfold channel, but if it is an iron, it will pass through the threefold channel. Okay. Oxidation to iron 3. We know iron 2 is transported in and out of a cell. It will definitely oxidize to iron 3. The oxidation to iron 3 is thought to occur at specific di-iron binding sites, which is known as peroxidase centers. What is a peroxidase center? Peroxidase center is that center at which oxidation of iron occur at specific di-iron binding sites. That sites is actually known as peroxidase centers, which is present in each of the subunits. We know in an apoferritin or a ferritin there is 24 protein subunits. Oxidation to iron 3 involves the coordination of oxygen, okay, coordination of oxygen, molecular oxygen, and an inner sphere electron transfer. Oxidation is loss of electrons. So there is an electron transfer occurs. Otherwise, there is a coordination of oxygen. Here you can see the structure of ferritin showing the arrangement of subunits. Here, a 24 subunits are here and that make up the protein shell. Okay. So the each subunit consists of a bundle of 4 long and 1 short helices with a loop that forms a section of beta sheet with a neighboring subunit. Okay. The mineral core is composed of hydrated iron 3 oxide with varying amounts of phosphate which helps to anchor it to the internal surface. The structure as revealed by X-ray or electron refraction resembles that of the mineral ferrihydrite, that is Fe2O3 9H2O, which is based on an hexagonal close packing array of O2 minus and OH minus ions with iron 3 layered in both the octahedral and tetrahedral sides. So in a ferritin there are a protein shell and also a mineral core. We know the protein shell consists of a 24 subunits, and there is a bundle of each subunit consists of a bundle of 4 long and 1 short helices, okay, with the with a loop that forms a section of beta sheet with a neighboring subunit, okay. Whereas in the mineral core is composed of hydrated iron 3 oxide. We know 4500 iron atoms are there in a mineral core, and it is composed of hydrated iron 3 oxide with varying amounts of phosphate, which helps anchor it to the internal surface. The studies shows that the mineral ferrihydrate, which is based on the hexagonal close packing array of O2 minus and OH minus ions with the iron 3 layered in both the octahedral and tetrahedral sites. This is about its structure. Here you can see the structure of ferritin and its outer or inner diameter. That is, here you can see that 12 nanometer is its outer diameter and 8 nanometer is the inner diameter. The graphic representation B, that is the graphic representation of ferritin subunit structure. We know there are 24 subunits are there and it is some more helices. And here the C structure, which represents the inter-subunit interfaces of ferritin, that is C3-C4 interface, C2 interface, that is the two-fold channel, and C3 interface, there is a pores through which the ions enter into the cell and out of the cell. Okay, its pore size is 3 to 5 angstrom. Okay, there is a C4 interface, that is a four-fold channel. There is also a pore which is hydrophobic one, whereas the C3 interface is hydrophilic one. So this pore is suitable for the metal ions transfer. What are the functions of ferritin? Here you can see the functions. Ferritin is present in every cell type that we know. It serves to store iron in a non-toxic form, to deposit it in a safe form, and to transport it to areas where it is required. We know functions of iron is a storage unit, that it's store iron in a non-toxic form. That is very important, and it is safe. Iron deposit in it is very safe, and to transport to various areas or various tissues where it is required. The function and the structure of the expressed ferritin protein varies in different cell types. This is controlled primarily by the amount and stability of the messenger RNA. The function and the structure of the ferritin protein varies in different cell types. That is controlled by the messenger RNA or mRNA. Free iron is toxic, that we know, all to the cells, as it acts as a catalyst in the formation of free radicals from reactive oxygen species via the Fenton reaction. Hence, vertebrates have an elaborate set of protective mechanisms to bind iron in various tissue compartments. Within cells, iron is stored in a protein complex as ferritin or the related complex hemosiderin. Here you can see a note of Fenton reaction. You can read it. Apoferritin binds to free the ferrous ion and stores it in the ferric state. Apoferritin binds to free ferrous ion and stores it in the ferric state. That we know, It will transport in and out of the cell as ferrous iron, and it stores it in the ferric state by oxidation. As ferritin accumulates within the cells of the reticuloendothelial system, the protein aggregates are formed as hemosiderin. The reticuloendothelial system means that is the RES removes the immune complexes from the circulation in healthy persons and is formed of phagocytic cells that are found in the circulation and in tissues. We know the aggregation of ferritin results in hemosiderin. Hemosiderin is toxic that we have already discussed. Iron in ferritin or hemosiderin can be extracted for the release by the RE cells, although the hemosiderin is less readily available. That removes the immune complexes from the circulation in healthy persons. Iron is released from ferritin for use by ferritin degradation, which is performed mainly by lysosomes. Iron is released from ferritin for use by ferritin degradation, which is performed mainly by lysosome. What is a lysosome? Lysosome is a membrane-bound cell organelle that contains digestive enzymes. So here you can see a table that contains the average human iron distribution. Hemoglobin is a protein that its function is plasma oxygen transport. The amount of iron is 2.6 gram, that is percent of total is 65%. Myoglobin muscle oxygen storage that contain 0.13 gram of iron. Transferrin is the plasma iron transport protein. It contains 0.007 percentage, gram iron. Ferritin is the cell iron storage, that is 0.52 gram iron stores. Hemosiderin cell iron storage, it contains 0.48 gram iron. Catalase is used for the hydrogen peroxide metabolism. It contains the amount of iron is 0.004 gram. Cytochrome C is used for the electron transport and it also contains 0.004 gram iron. Other such as oxidases, other enzymes which contain 0.14 gram of iron. So this is the average human iron distribution in various proteins. And here the protein's functions are also shown here for your attention.